Yes, sir.
Okay.
Yes, sir.
So welcome
Yes,
back
sir.
to the second session of this refresher course. Once again, I thank the Academic Staff College of the University of Mysore and Professor Janardhana Madam, the coordinator of this refresher course, for inviting me to address on another important topic. It is very important from the point of understanding the structure and functions of this RNA molecule, and also a lot of research work is going on currently on this molecule on various aspects of the RNA world. And this is one such a small topic, but it has been already introduced in PU college students. For PU college students also, this topic, viroids, has been already introduced. And at the degree level also, we have, while framing the syllabus, we introduced this topic, realizing the importance of the topic. Wherever necessary, this particular topic has been, incorporated with the intention that our student must learn, because this is one of the topic which is going to help us in understanding the structure, mechanism of disease caused by this RNA molecule, though it is appears to be recent for all of us, but the study began as early as in 1922. So let me come back to the topic. Before getting into the topic on infectious RNAs, let me briefly introduce the crop on which this particular problem was detected. So as you all know that potato is not our vegetable crop. It is a South American crop, but today it is the #1 staple vegetable crop all over the world. The botanical name, as you know, Solanum tuberosum, and belongs to the family Solanaceae, and one of the staple food crop of the world. And it is a native of South America. Specifically, it was domesticated in a region called Peru, and later it was introduced to other parts of the world. In potato, there are about 5,000 potato varieties all over the world. You may not believe that.
Yeah.
And the Spanish introduced this potato crop to Europe. Several attempts were made to introduce this potato crop to Europe. They failed in the beginning, but later somehow they were successful in introducing the potato crop to the Europe in the second half of the 16th century. And because of similar weather conditions in South America and also in Europe, this crop has very well adapted to European region, and it is a major vegetable crops in Europe and rest of the world. And today, worldwide, China is the #1 crop in terms of production, followed by India. Way back in 1842 to 45, when this potato crop was grown very well in European continent. In 1842 to 45, all of a sudden, a disease appeared that was known as late blight of potato. It was appeared in Ireland, the part of United Kingdom, and devastated the vast areas of potato crop cultivation, and also in other parts of Europe. The fields, potato fields, which promised bumper harvest, they all transformed into a rotten field within a span of 15 days, and all the tubers were spoiled underground soil. Because people were dependent on potato as their staple food crop, the hunger and dysentery and typhoid fever spread among the farmers. And during this particular period, 1.5 million Irish people died from hunger, and same number of people migrated to other parts of the world. So if you just Google it in the internet, if you type it Irish famine, you will get a detailed account on the Irish famine, what was responsible for Irish famine, and how this disease, late blight of potato disease, destroyed the potato cultivation pattern in Europe, and how this was responsible for starvation and death in European region. And 1845 to 49, again the disease started appearing in the subsequent years. and they could not able to identify the causative agent of the disease, and as a result, starvation gripped the whole Europe, and as I mentioned, 1.5 million people died due to just starvation. The British royals who brought an eviction act during the same period, because they were all the peasant, particularly farmers who were growing potato crop, were actually poor, and they were all bonded laborers. And during that particular period, the British royals brought an eviction act, and the tenants were thrown out of their homes in Ireland for not paying the rent. And finally, for these people, the help came from United States of America and also Canada. During that period, millions of people left homeland Ireland and took shelter in America and Canada. The Ireland population before 1942, it was about 7 million, 14 million, and reduced to 7 million. And these statistics tell about the severity of late blight of disease. This is the picture just showing the tenants were thrown out of their homes in Ireland for not paying the rent by British royals, and the homeless took shelter on a countryside, roadside, and nearly 1.5 million people died, and almost same number of people migrated to USA and Canada. And while migrating from Ireland to other part of the world, USA and Canada, many people died, particularly while boarding to the ship. So to remember that, very inhumane attitude of the people in those days, the remembrance of that great famine, 85 to 45 to 49, which saw the population of the country hallowed through the death and immigration, a beautiful Irish memorial was constructed in Dublin, and that speaks about the cruelty of the humankind and the deadly mix of ignorance when late blight disease struck the potato crop in Ireland. And it was almost 15 years later, the disease was appeared in 1845, and it was in 1861, a German botanist, Anton de Bary, who did a simple experiment. He was the one who proved that a late blight of potato disease was caused by a fungus known as Phytophthora. That means Phytophthora meaning, phyto means plant, Phthora means destroyer, a plant destroyer. This is how the fungal name Phytophthora was given, and it was responsible for the disease. And therefore, a systematic study of plant diseases began. And hence, Anton de Bary is called as father of plant pathology. He is one of the important discipline in botany. And now the discipline botany has identified several different types of agents responsible for causing diseases in agriculturally, horticulturally important crops. These can be divided into non-parasitic diseases and parasitic diseases. Parasitic diseases are those which are caused by the living organism, biotic agent, such as bacteria, fungi, algae, protozoa, angiospermic parasite. Even higher plants do parasitize other plants, and even nematodes and insect, phytoplasmas, small L-shaped bacteria, and later viruses and viroids were also known to cause diseases. They have been identified. In addition to that, there are non-parasitic diseases. That means the unfavorable environmental condition, such as excess and deficiencies of minerals in the soil, toxicity of certain chemical air pollutant, extreme environment such as high temperature, high moisture content, high relative humidity, or air pollutant, excess pesticide, they do cause diseases, and they all grouped under non-parasitic diseases or diseases caused by biotic agent. When it comes to biotic agent, there are so many living organisms. Today, I am going to talk about the last category of causative agent which cause diseases in plant responsible for significant economic loss. They are viruses and viroids. Let us try to understand one or two slides. What are viruses? You know that viruses are considered a disease-causing agent after the discovery of electron microscopes. Viruses were submicroscopic in nature. They were detected in plant tissues, and they were considered as agents of plant diseases in plant system. Viruses are nothing but nucleocapsid particles. That means they're made up of nucleic acid. So in the center, there will be nucleic acid surrounded by a proteinaceous coat. We call it as a capsid. So in simple definition, they are referred to as nucleocapsid particles. They consist of small piece of nucleic acid, either RNA or DNA, never both. So remember this, okay? In the central portion of the virus particle, we have RNA or DNA, but never both. Surrounding this nucleic acid, we have a protein coat or lipoprotein coat. We call it as a capsid. When you say virus, these two important particles are very important. One is the nucleic acid, and the other one is proteins. Very important. So nucleic acid sequences range from 1,000 nucleotide to 250,000 base pairs. So this is the length of genome size of the virus particle, which are known to cause diseases in plants. So to go further into the virus definition, they are ultra microscopic in nature. We need an electron microscope only to identify them, and they are acellular in nature. That means they never contain a protoplasm or a cytoplasm and a cellular organelles. So that's why they are called acellular in nature. They are non-culturable, very difficult to culture on media, inanimate media, and they are obligate intracellular parasite. They live only in the inside the living cell. So credit goes to Adolf Meyer, a Dutch botanist. As early as in 1886, he observed that tobacco mosaic disease in tobacco plant. That was the starting point for the identification of the virus. And later it was Dmitry Ivanovsky, a Russian botanist, in 1892, stated that it is tobacco mosaic disease is highly infectious. That means it spread from infected plant to healthy plant. Later, it was the turn of M.W. Bejernik in 1898, called this infectious agent as Contagium vivum fluidum. That means living infectious fluid. He coined the term virus. Okay. So therefore, later it was in 1936, W.M. Stanley purified this infectious agent in tobacco mosaic disease in tobacco, and purified this virus particle, and he named them as tobacco mosaic virus in 1936, for which he was awarded Nobel Prize later. So there are basically three kinds of viruses are there. We, animal viruses in which DNA is the genetic material, and we have plant viruses wherein RNA is the genetic material. In addition to that, we have bacteria infecting viruses. They are called bacteriophages, in which DNA is genetic material. Though it is a simpler classification, there are exceptions to that. We have HIV virus, wherein RNA is the genetic material. Similarly, in Gemini viruses, they are plant infecting. We have a DNA is the genetic material. So okay, we have a MS2 and Q beta have RNA as the genetic material. Okay. And look at this. This is a simple animal virus influenza. We have, as I mentioned, viruses are made up of nucleic acid. Look at this. Centrally placed nucleic acid thread, externally surrounded by a protein coat. In case of viruses, there will be one more protective layer called lipoprotein layer. In case of this HIV virus, we have a nucleic acid RNA, centrally placed nucleic acid RNA surrounded by the protein coat. In turn, this protein coat surrounded by a lipoprotein membrane, which is of human origin. Okay, it is any virus for that matter. Look at that plant virus. We have a RNA molecule in the center, externally covered by a protein coat. So viruses means a nucleoprotein cap. Nucleoprotein particles. So centrally placed nucleic acid surrounded by the protein coat is the characteristic feature of viruses. We thought that these viruses, responsible for causing diseases in many agriculturally and horticulturally important crops. No one ever thought that only RNA, only DNA alone able to cause diseases in plant. So it was in the 20th century we discovered that only RNA molecule, ribonucleic acid, is known to cause disease in plant. So we call them as infectious RNAs. So scientific studies in the 20th century proved that the small piece of just RNA is smaller than viral RNA. As I mentioned, viral RNA will be 1000 nucleotide to 250,000 nucleotides. But here, look at this. Viral RNA is just 250 to 500 nucleotide base pair nucleotides. It is a very small RNA and can cause plant diseases in plant. We call them as infectious RNAs. So today, this is my topic of today's presentation, infectious RNAs, molecular parasites of crop plant, on which I am going to discuss more on the very interesting aspects of these infectious RNAs. So let us first discuss what are viroid. So these infectious RNAs are otherwise known as viroids. I will come to that later. So these infectious RNAs or viroids can be defined as they are very small, single-stranded. So RNA always single-stranded, okay? And they are circular in nature. When they are present inside the host cell, they are circular in nature, low molecular weight, naked RNA. Why we call them as naked RNA? Because there is no protein code, only the small piece of RNA, which ranges from 250 to 450 bases. Look at this, 250 to 450 bases, able to cause diseases in plant. We call them as infectious RNAs or viroids. So hope you are cleared with the definition. And the name viroid means virus-like particle. It is not virus, but we call it as a viroid because it is a virus-like particle. Okay, so that is why the name viroid has been given the definition. These are all naked plant-specific infectious RNAs. That means they infect only plant cells. Okay. So there are some exception very recently that I will talk about it later. Is there any, RNA, small RNA able to cause human diseases? Answer is yes, but also no. That I will tell you later. As on today, only RNAs, infectious RNAs, able to cause diseases only in plant. So that's why they are nothing but a naked plant-specific infectious RNA, tenfold much smaller than smallest viral RNA, okay, viruses. So they are called the viroids. The viroids are now considered as the lowest step in the biological scale. They are the molecular parasite at the frontiers of life. So far, we believed that viruses were considered as the lowest, smallest particles known to cause diseases in plants and animals. But now the discovery of the small RNA showed that they are the much smaller than viruses and they are considered as molecular parasite at frontiers of life. Let us discuss further. What are the important characteristic features of viroids? How do you identify it is a viroid from viruses? So as I mentioned, viroids are free from proteinaceous capsid, protein capsid. That is one thing. They are made up of only RNA, small RNAs. So therefore, these are the three important characteristics by which viroids, small RNA, infectious RNA can be distinguished from viruses. One is they are very small, circular in nature without a protein code. All viruses have protein code. All viruses have protein code or lipoprotein code. If it is a virus is animal virus, we will have lipoprotein code. But look at this, viroids, they are small circular RNA without a protein code. They lack genes to code for proteins. Unlike viruses, viruses do code for one or two enzymes, which helps them in replication. But viroids, even those genes are also, which code for enzymes are proteins are lacking. So lack genes to code for proteins. Some of the viroids, these RNAs have catalytic property, self-cleaving ability. self-cleaving ability. Without the assistance of host RNA, they can multiply themselves. They are called ribosome activity. They replicate themselves without the host enzyme machinery. So these are the three important characteristics by which we can distinguish the virids from viruses, infectious RNAs from viral RNAs. So the study of this virid, study of this infectious RNAs began as early as in 1922. Potato spindle tuber disease was first described in 1992, and commercial potato crop cultivation and potato breeding lines in US was seriously affected by this particular type of disease until 1967. Look at this, 1922, the disease was identified. Till 1967, potato spindle tuber disease was universally, people believed that the disease was caused by viruses. So therefore, all the scientists, even at the US Department of Agriculture, they started investigating the presence of viruses or associated viruses with the potato spindle tuber disease. Look at this. This is a healthy tuber. This is a spindle-shaped tuber, potato tuber. If there is an infectious agent enters the plant body, all tubers will become spindle-shaped. And as a result, the productivity and the overall commercial production of potato tuber seriously affected in US. Later, it was in 1971. It was in 1971, the term virid, in fact, was proposed by a T.O. Daner. A T.O. Daner was an agriculture scientist, rather plant pathologist at the US Department of Agriculture, USDA. In 1971, he identified these small RNA agent from viral RNAs. Small viral RNAs from viruses able to cause diseases in potato and other crops. He identified as the causative agent of potato spindle tuber disease. So the small RNAs which are distinctly different from viral RNA. So he named them as virid, and today there are more than 32 different types of virids, and more than 160 variations have been detected and are known to cause diseases in economically important crop plant. So remember this, credit goes to T.O. Daner, an American plant pathologist who identified this agent as virid, and they are smaller than viruses, able to cause diseases in plants. So when T.O. Daner proposed that these small infectious RNAs, small RNAs responsible for diseases in tomato, at that time, the well-known virologists, they never accepted the view that small RNA is able to cause diseases in plant. However, with many debates with T.O. Daner in the International Congress of Virology, Andrew Love, he was one of the most respected authorities in virology. He was convinced the existence of virid, and he finally said that the time has come for accepting the virids. So till that time, we were all believed that viruses were the submicroscopic agent, ultra submicroscopic agent responsible for causing diseases in plant. But in 1971 afterward, when T.O. Daner suggested that there are small RNAs which are much 100-fold much smaller than viral RNA able to cause diseases, and in the International Congress, it was accepted that yes, the small RNAs present and they are known to cause diseases. Then systematic study of the virids began from 1971 onwards. They are currently regarded as the lowest step of the biological scale, the molecular parasites of frontiers of life. Look at this. The virid which was associated with potato spindle tuber disease was finally discovered. It was measured that it was about, the size was about 250 to 450 base pairs. Look at this, PSTVD. This is an entire picture. It is an E. coli cell. Okay. So when compared to the Escherichia coli, look at the size of PSTV, potato spindle tuber wider. It is the smallest one. Okay. And we have a tobacco mosaic virus. Look at this. Okay. This is a tobacco mosaic virus. This is a T4 phage. Okay. This is a vaccinia virus. So it is just PSTVD size compared with the other microbes. That's why I told you it is the smallest microscopic, submicroscopic living, RNA particle able to cause disease in plants today. So spindle tuber, later the potato spindle tuber viroid was identified and it was sequenced and it was named as PSTVD. For all viruses, we call it as potato spindle tuber virus. If it is a virus, we use this capital V, virus. But if it is a viroid, small d we add, okay? Viroid, PSTVD. For any viroid, if there is a VD, that means it is a viroid. That means it is a small RNA, which is less than 500 base pair, known to cause diseases in plant, and we can name it as on the basis of host in which it caused disease. Potato spindle tuber disease, viroid, has been the first independently replicating subviral pathogen identified, and it has been very well characterized. Look at this, look at this. Okay, this is the structure of potato spindle tuber viroid, and it is a secondary structure proposed from time to time, and it has been very well studied viroid in the plant pathology. Now I will talk about a little bit more on this. So look at this. When there is a infection by potato spindle tuber viroid, okay, look at the size, okay. This is an uninfected potato tuber. We have, I told you, more than 160 variants of potato spindle tuber, viroids have been identified. Each one has their own characteristic, okay. With successive generation, the size of the potato tubers, gets reduced because of infection by these small RNAs. And there will also be a reduction, malformation, various degrees of growth reduction, and cracking. There will be cracks on the tubers. These are all the important characteristic features by which we can identify the associated viroids in any potato crop. Look at this. This is another picture just to impress upon that how small RNAs, though they are very small, able to cause such a drastic diseases in agriculturally important potato crop. And this is RNA preparation. Finally, they isolated the RNA and they just compared. This is the electron micrographic picture of look at the RNA, okay, small RNA, PSTV, PS potato spindle when compared to the T7 DNA, CT7 DNA, okay, it is just to give a comparison how potato spindle tuber is, how small it is, okay, just to show that. Coming to the genome size of viroids, as I mentioned, they are very extremely small. Viroid genomes are extremely small and covalently circular. When they are present in the cytoplasm, they take the shape of a circular in nature. They are about 250 to less than 500 nucleotide sequences. So only that much. Genomes adopt a rod-like structure. When you isolate RNA from the host cell, they take the shape of a rod-like appearance. Secondary, structure, rod-like appearance. Otherwise, they are circular, covalently circular when they are present inside the living cells. The rod-like structure of viroid contains 5 functional domains have been identified. So these RNAs have been completely sequenced. Complete data is available with us. Based on that, secondary structure has also been constructed. Look at this. Types of viroid. Based on the secondary structure of the viroids, they isolated 2 types of viroids have been identified. One is rod-shaped viroid. The other one is branched viroid or hammerhead viroid. Hammerhead, because the viroid will take a shape of a hammerhead. Hammerhead. So that is why the name. There are only 2 types based on the structure. One is rod-shaped viroid and the second one is hammerhead-shaped viroid. When they are present inside the living cell, both are circular in nature. Single-stranded circular RNA. Okay. So remember this. These viroids have been, ultrastructure of viroids have also been studied. All virides have 5 domains. What are those 5 domains? One is the upper one is the rod-shaped viride. The below one is the hammerhead shape. Look at the shape. It looks like a somewhat hammer, okay, hammerhead. So that is why they are called hammerhead shaped viride. 2 types of viride. Whether it is a hammerhead shaped viride or a rod shaped viride, the 5 regions are very common. We have in the central C region, CCR region, central conserved region. In all viride, this region is highly conserved. On either side of this central conserved region, we have pathogenicity region and variability region. Pathogenicity region believed to be involved in causing diseases in plants. Various types of symptoms that are expressed in host plant is actually due to expression of certain sequences which are present in the pathogenicity region. The variability of virides, I told you more than 160 different types of viride variants are there. That variation that we can see only in the variability region. On either side of the pathogenicity and variability region, we have a right terminal end and we have a left terminal, right terminal end and we have a left terminal end. So this is simplest structure of a viride. They are very small basically, but when they take the shape of a rod, they can be, you can distinguish them into 5 major domains: central conserved region, pathogenicity region, variability region. On either end, we have a left terminal end and right terminal end. So it is also with other hammerhead shaped viride. So the 5 structural domains include conserved central region, CCR region, pathogenicity domain, variable domain, left terminal repeat, right terminal repeat. So similarly, the Y-shaped or the hammerhead shaped viride do have the same structure. So all the virides which have been characterized, isolated from different agriculturally important, horticulturally important cropland can be classified into 2 major classes. One is called Paspiviridae. This is one particular family. The other one is called Avicen viridae. So only 2. Okay, in Paspiviridae, all contains rod-shaped viride and central conserved region is the main important domain. So this we look at this viride. PSTBD, potato spindle tuber viride, coconut kadang viride. Okay, so like this, half stunt viride, coleus bloomy viride. So these are all these how virides have been abbreviated. So this is a standard abbreviations we have to use when we write, scientific papers and all. So we have a hammerhead viride comes under Avicen viridae. They are the virides looks like a hammerhead, okay, or a quasi rod shape. So the best examples we call Avicen viride, avocado sunblotch viride. This is the best example for rods, hammerhead shaped viride. The other one is peach latent mosaic viride because the secondary structure of these 2 virides looks like a hammerhead. So hope this much is clear to you. Look at this. Okay, these are the 2 important families of viride. Family Paspiviridae, Avicen viridae. These are the important virides that have been identified, characterized. They are known to cause serious diseases, which are responsible for significant economic loss in croplands. So that's why a lot of work has been done on these things. So as I mentioned, potato spindle tuber viride, coconut kadang, these are the important diseases, including citrus exocartis disease. It is there in India also. Chrysanthemum chlorotic mottle disease. This is also a very important article, floricultural crop, chrysanthemum. And now this is affected by the viride, chrysanthemum stunt viride. Okay, so these are all very common. I will tell you, even mulberry plant and grape vine, even in India, they are also infected by viride. I will give you 1 or 2 examples later. How to detect these small virides? One is by morphological methods. So visual methods. So we call them as symptomatology. How a doctor identifies, particular disease to a particular causative agent. Similarly, we can, based on symptomatology, symptoms, we can identify. But there is a overlapping symptom. Virus, phytoplasma, and virides. They are known to cause similar type of symptoms. Very difficult. Only expert pathologist can identify a virus disease and distinguish the virus disease from viride disease. Otherwise, there will be overlapping symptoms. So therefore, what they have done later in the beginning, they isolated the RNA content, and based on the RNA content, they analyzed the RNA analysis. Based on that, they could be able to distinguish two types spectrophotometric analysis revealed the presence of two peaks, that is host RNA can also be distinguished from the other viral RNA and virad RNA. So later electron microscopic observations also revealed the presence of virides, and later bioassay indicator plant. We can also conduct certain experiment on sensitive plant. There are some plant, when you inoculate these virides, they induce disease. Based on the appearance of the symptoms, you can identify whether it is a viral disease or a viral disease. So such assay is called bioassay method. The other one is slot blot. Now molecular biology techniques are available. By using a specific probe, you can identify, whether it is a virad or virus. So another important method is a two-dimensional page analysis. That means when I said RNA isolation, when you isolate RNA, you will get tRNA, mRNA, all host RNA will also be isolated. But when you do two-dimensional RNA analysis, polyacrylamide gel electrophoresis will reveal you the presence of another small RNA that can be considered as very 100% perfect method to identify the presence of small RNA. Okay, that will 2D page will distinguish the presence of the small RNA. So now molecular biology tools like RT-PCR, because it is the agent is a RNA, we have to convert the RNA into DNA first and then do the PCR method. That's why we call reverse transcription PCR method. This will give you 100% conclusive result whether the virids are present associated with a particular disease or not. Once you do that, the PCR products can be subjected for cloning and sequencing, and sequence analysis and phylogenetic analysis definitely will take you to identify the correct virid that is associated with particular plant disease. So this is just a 2D fudge showing the presence of small RNA, which will distinguish from the host RNA. Yeah, we can distinguish the presence of small RNA from the host RNA. As I mentioned, we have a bio-indicator plant, like Cucumis sativus, a tomato plant, a root jar variety of tomato plant, and then cucumber, so Cucumis sativa, Lycopersicon esculentum, Nicotiana benthamiana. It is a all solanaceous plant and one cucumber plant. We can use them in the laboratory. Okay, so at the two-leaf stage, if you isolate the RNA from the infected plant, inoculate with the help of carborundum. How we inoculate to detect the viruses in plants? Similar method. In a two-leaf stage, we have to inoculate these plants with a small RNA with a phosphate buffer pH having 7. You rub them and then wash with a sterile water and allow them in a greenhouse condition. Look at this. This is a healthy plant and this is a healthy plant. Look at the reduction in growth of these plant after 30 days. That is a clear indication. It is a clear indication that the virids are present. One of the distinguishing feature of virid is that they will never cause yellowing, chlorosis. Okay, plants will be greenish only. Plants will be greenish, but in case of viruses, the stunting is followed by chlorosis or yellowing of plant is always associated with the viruses. But in case of virid, there will not be yellowing. So based on this, we can distinguish at the very young stage that the virids are present. So these samples can be used further to detect by using RT-PCR. You can confirm that virid is really present or not. This is how we can propagate, virids in indicator plant or bioassay plant. We can always keep them in the laboratory, and whenever you want, you can detect them, conduct the experiment on whatever you want. Then how do they replicate? Because I mentioned that there are no genes are associated to code for proteins or enzyme. Then how do they replicate? Unlike viruses, virids do not code for any proteins or enzymes. So virions are very clever. Virions replication mechanism uses RNA polymerase II. It is a host enzyme that normally associated with synthesis of mRNA. The same host mRNA, RNA polymerase II is utilized. So this polymerase II synthesizes new RNA copy using the virions RNA as template. So some virions, as I mentioned, for example, hammerhead virions, which have catalytic property, they have autocatalytic property, they self-replicate themselves and then ligation. So as a result, they multiply themselves. Okay. Only rod-shaped virion, they have used the host RNA polymerase II for multiplication. So there are two types of virions, RNA molecule, small RNA molecule circulate in the plant system. One species, circular RNA, we call it as a plus strand. The other one is a minus strand. So all these, while replication, the plus strand acts as a template. So nucleus encoded polymerase, polymerase II enzyme synthesizes, use this plus strand as a template and synthesizes a new copy of the RNA molecule, and then linear RNA is synthesized, and with the help of RNA ligase enzyme, these linear molecules undergo circularization. It becomes circular, and this we referred as minus strand. Later, this minus strand acts as a template for synthesis of another strand, plus strand of virion. Again, nucleus encoded polymerase enzyme, RNA polymerase II synthesizes a new strand of RNA, and then it gets linearized, and the RNA ligase circularized it, and you will have plus virion strands are synthesized. So both plus and minus strands
Hello, sir.
do circulate.
Sir.
Yes. Hello. Hello, please.
Sir, is there any genetic change in the genetic makeup of plus strand and minus strand?
It is almost same. It is a replica of the same. So the plus strand is known to be present only in the nucleoplasm, and the minus strand is known to circulate in the other part of the plant body, including the phloem. You know that all viruses, they are present only in the phloem sieve tube. Similarly, virions are present only in the phloem sieve tube. When you isolate, you will get a minus strand, most of them. So what is the exact mechanism? Why virions have retained plus strand in the nucleoplasm, and why the minus strand circulate in the plant tissue, we do not know. But this is again hypothetical model, okay, that has been proposed how PSTVD replication takes place inside the plant nucleoplasm. This is the same picture, okay. This is the same picture in a to explain you much better. So what are the important symptoms that are produced by these small RNAs by which you can distinguish from other symptoms which are caused by other biotic agent? Symptoms of viral infection include stunting. Of course, viruses also cause stunting, leaf epinasty, sometimes distortion, leaf distortion. Okay, so leaf lamina gets distorted, fruit distortion. The fruit size gets reduced, and same color breakdown. The flower color will be different, leaf color will be different, stem and leaf necrosis do takes place depending on the type of host, and later even the death of the whole plant takes place. And this is often dependent upon environmental condition. So, but the stunting and leaf epinasty and distortion of floral buds, these are all the important characteristic features of viral symptom. But of course, as I mentioned, these characteristic symptoms overlap with the viruses and as well as phytoplasma symptoms. Only expert eye can distinguish the symptoms which are caused by the virions. Then how do virions induce disease? We do not know. The little is known about the mechanism by which wire rats cause disease in plant, but it has been speculated. I will tell you later. The cell wall malformation takes place, membranous bodies with distorted organization, chlorotic disturbances in the subcellular level, changes in the expression of pathogenesis related protein. The host proteins gets synthesis of host proteins also gets affected. Alteration in the hormones have been detected in many host pathogen interaction. Certain metabolites at the biochemical level, all these changes have been detected. But the exact mechanism by which wire rats RNA, the small piece of RNA induced disease has not been perfectly identified. And how wire rats damage the plant is still rather mysterious. It appears that wire rat and its replicative intermediates, maybe minus strand or plus strand, they trigger the RNA interference, something like a gene silencing mechanism. Gene silencing mechanism. Gene silencing mechanism is nothing but that when transcription occurs, we have a template DNA. DNA gives rise to mRNA during transcription. So the mRNA at the mRNA level, the formation of mRNA gets affected. As a result, functional protein formation, gets affected. So the people, expert stated that these small RNA interfere with the mRNA transformation. So this results in the production of short single-stranded RNA molecule. We call it as a virus RNAs or viride RNA or viride short RNA. These viride short RNA likely to interfere with mRNA formation of functional genes and the resulting in gene silencing of host metabolism. So that is the proposed. This is again a proposal. The virides, they do produce the small RNAs. We call them small interference RNAs. These small interference RNA will interfere with synthesis of mRNA. The mRNA gets degraded. So host mRNA gets degraded. As a result, the symptoms will result. Instead of producing a very important molecule which is necessary for normal functioning of the plant body, as a deficiency of that, symptoms will appear on the plant body. So this is a proposal that has been proposed as far as the disruption of cellular functions of host system by RNA. So this is the same picture. This is how the viride short RNA likely to interfere with mRNA, result in the disease formation. As I mentioned, the virides are known to circulate from cell to cell through plasmodesmata connections. But plus strands of virus are known to be concentrated in the nucleoplasm. Only the minus strands are known to be circulated in the plant body. And long distance travel is through phloem, particularly sieve tubes. You know that plants are consists of xylem and phloem. The phloem consists of phloem sieve tube, and these virides are known to be present in only in the phloem sieve tube, and particularly the minus strands. So several studies have been proved that the plus strands known to be more so in the nucleoplasm and minus strands are known to circulate in the plant body. So what are the important crop plants that are affected by virides? That include both monocot plant and dicot plant. Both are affected, and they are known to cause serious diseases, both in agriculturally important crop as well as in horticulturally important crop. Look at this particular map, geographical distribution of virides. So throughout the world, almost throughout the world, even in India also, we have detected good number of virides, including Karnataka. We in our lab also we have detected. I will tell you some of the interesting aspects of that. All over the world, these virides are distributed. Then how do they spread? How do they transmit from infected plant to healthy plant? They are known to be transmitted through infected vegetative propagation unit. For example, potato. Potato is propagated through tubers, and therefore they are likely to be transmitted from next crop through tubers, which are used for propagation. Then mechanical transmission is also possible, and then grafting. When you do grafting with using certain tools, if the tools are infected, if you use the same tools without disinfection to graft the other healthy plant, the viruses may get transmitted. So therefore, agricultural implements do contribute the spread of the viruses. Infected seeds and pollen grains, they are likely to contain viruses, and therefore, through them, the viruses gets transmitted. Phloem sucking aphid, because the viruses are present only in the sieve tube element. There are many aphids and insects. They suck the juice from the plant through phloem element. So when they suck, the viruses are getting into the insect. When they feed on healthy plant, there is a chance that the viruses are likely to be introduced into healthy plants. So vegetative propagation, mechanical transmission, and pollen grain, it has been very well proved in different host pathogen interactions. Then how to manage these viral disease? As on today, for viruses also, there is no management method. This is, chemical, antiviral chemical, we can use them to prevent viruses. Similarly, for viruses also, there is no solution. There is no chemical viricide that is available to control the diseases. Planting only the virus-free plant, diseased material or propagation material is the only solution at the present. That is why we have to be very careful about transmission of viral diseases. Use of resistant varieties. So people have identified certain varieties are resistant. Okay, such varieties should be used. Eradication of the infected trees. How we do for viral diseases? So phytosanitation. Okay, raguing. Burning by killing. Okay, infected viral plant should be killed immediately so that inoculum spreading of the inoculum can be avoided. Similarly, eradication of the infected trees and plant, the insect, because the viruses are also known to be transmitted through insect. By controlling the insect, we can control the chances of spreading of the disease. The best method, phytosanitation. So in the field, we have to follow phytosanitation so that we can control the disease spread to other areas. So coming to specific examples of viral disease, I will take another few minutes to dwell upon why viral diseases are very, very important. Look at this particular picture. It is a coconut kadang disease. This field depicts the disease caused by the viral. It is known as coconut kadang disease. This is a picture of 1929. 1929. It is a coconut garden in Philippines. Coconut garden in Philippines. You know that the Philippines, they have a very good coconut gardens were there. And coconut kadang disease was a serious disease that affected the coconut gardens in Philippines in 1927 to 28. It was first reported, this disease, in 1927 to 28. By 1962, all 25 million palms died in Philippines. You can imagine, 25 million palms. So far, 40 million coconut palms have been killed since it has been discovered. Okay, and economy of the Philippines, a small island, it was jeopardized by this because they were all dependent on this important, horticultural crop. Now, the Philippines economy now dependent on the tourism. Look at this picture, the same picture, look at the leaf, infected leaf and the healthy leaf. So when there is a viral infection, the leaf will become yellowish. By fifth year, it will take 5 years, maximum 5 years, all coconut trees will die. Okay, the fruit size will get reduced and poor yield. Okay, and finally, within 5 years, all the coconut plants will die. It was Randall, he was one of the authority. He worked on coconut kadang disease, and he stated that it takes minimum 5 to 6 years for the death of entire coconut trees. Look at the same. Okay. So later, it was in 1981. 1981, the virid was isolated. The genome of the virid was sequenced in 1981, and the size was 246 RNA molecule. a naked RNA molecule. It was just 246 nucleotide molecule, and it has a central conserved region of 44 nucleotides, and it has the viride has been named as CCR, CCCVD, coconut kadang kadang viride. It has a number of molecular forms are there, and this viride is now known to, this is the RNA preparation. When they isolated the RNA and they electrographed under electron micrography, can you see the small circular RNA? Okay. These are all the virides, small RNA preparations, okay, showing the presence of virides in the RNA sample isolated from coconut kadang kadang disease from coconut. And later, by 1988, 98, the coconut kadang kadang disease was also, I noticed in Tinangaja region of the other part of the world, and they have isolated another virus called coconut Tinangaja virus. CTIVD. Okay, so this is another viral disease. And later, I have already given potato spindle tuber virus known to cause disease in potato, but it was also reported in tomato later. Same virus, it has no, because both belongs to the same family, Solanaceae, and later it was detected. Look at the leaf color, okay, leaf color and size of the tomatoes, okay. So they get affected. When there is an infection, PSTVD and tomato, the leaf size gets reduced. The plants will be greenish and the flower parts will be affected. No flower setting, fruit setting, and as a result, they will not produce any potato fruits, and yield will be 100% affected, and productivity of tomato. It is an important vegetable. Even India also we have identified this virus. Look at this. Okay, so symptoms, if you compare, they all looks like a virus. Looks like a virus. So that's why very difficult. Only molecular biology tools will identify whether it is a small RNA viride or virus. It is a tomato chlorotic dwarf viride. TCDVD, okay, tomato chlorotic dwarf viride. The plants will become very dwarf in nature. It has been reported, even in bell pepper, pepper chart fruit. Look at the size gets reduced. If it viride if it infects, this chili plant, the size of the fruits gets reduced significantly. It has been reported in apple. Look at the apple, Indian apple scar skin viride. The apple quality, particularly the skin color gets affected. No one will buy if there is a scars or cracking on the apple fruit. Lot of deformation. Okay, the fruit quality is severely affected, and therefore no marketability, market value of the product gets affected. So that's why I told you both the viruses are causing serious disease both in agriculturally important and as well as horticulturally important crops. Again, for apple scar skin virus on apple. And apple dimple fruit viride. Sometimes we do buy them. Okay, some of the apples which have a deformation, without knowing that they are all infected, we do buy them. And this is a ADFVD symptoms, and it has been also reported in grapevine. Even in India also, look at the small yellow pecks. These are all important characteristic feature. Leaf size gets reduced. Okay, leaf size gets reduced and yield will be affected. So it is also already been reported in grapes and in citrus exocartis viride. Indian Agriculture Research ARI, people have isolated this particular viride also, is citrus exocartis viride. The size of the citrus fruits gets affected, and even there will be a cracking on the bark. Cracking on the bark is an important symptom of citrus exocartis virus, CEVD. Look at this, bark cracking, bark splitting is an important symptom of citrus trees. And we have citrus bent leaf viride, another symptoms. And as I mentioned, chrysanthemum chlorotic viride, dwarf viride, is an important floricultural crop. So even in India, we have isolated the viride, and it is known to reduction of flower size. Chlorosis gets induced. Look at this. If there is a stunted appearance, they will never flower. They will not give you any flower. Even if they are produced, the flowers will be very small, and the quality of the flowers gets affected. So chrysanthemum stunt viroid. Columnar latent viroid, CLVD, okay, is another important viroid, and avocado sunblotch viroid. Avocado sunblotch viroid, and look at the fruit quality, how it has affected due to the symptoms. Peach latent mosaic viroid, even in cucumber plant, cucumber, the size will be reduced, okay, because of infection. And very recently, you must have seen this plant in most of the horticultural, nursery. Okay, it is called coleus plant, coleus bloomy. Okay, coleus bloomy. This in this particular plant, very recently, coleus bloomy viroid has been isolated, and this variegation, whether it is due to viroid or not, we don't know, but people suspected that the variegation is associated with imbalance in the normal physiology of the plant system. This is, we do not grow this crop, which is called Humulus lupulus. But this plant produces a female inflorescence. We call them as a strobilus. And these strobilus contain very important, beer flavoring and stabilizing agent. This is the one. Female flowers or strobilus are used in beer production. The characteristic color, taste, and stability of the beer depends on these female flowers produced by the hop plant, the Humulus lupulus. This particular plant and garden, particularly in Poland and Slovenia, they all affected, and now half-stunt viroid. All infected plants will be stunted in growth. The moment they become stunted, they will not produce these female flowers or strobilus, and therefore now in European countries, the beer production is affected due to this half-stunt viroid, HSVD, half-stunt viroid. HSVD has also been detected in mulberry. So, mulberry leaves, very important in sericulture, moriculture, okay. And leaf quality is very important. Cocoon production, cocoon quality, cocoon size, they all depend on quality of the leaves that we feed to the Bombax mori, silkworm. And now these leaves are getting affected by same HSVD, the viroid which is known to cause half-stunt viroid, that has been reported even in India. I will tell you the story of that. Okay, so now leaf quality is affected, so and it is going to affect the sericulture. So is there any viroid, that means small RNA, able to cause disease in humans? So that is the question. So very recently, we have a report that hepatitis delta viroid, HDV, hepatitis delta virus, which is similar in many characteristics, which resemble the viroid-like properties and may be related to viroids, we do not know. It has small circular RNA genome, and that's why it is called HDV. It is often called as viroid of animal. This is the only viroid. This is the only viroid that has been isolated and known to contain small RNA, able to cause diseases in plant. But we cannot treat it as viroid because of certain reason. Because this virus need a helper virus is required for self-assembly. And in this study, in this way, the HDV differ from plant viroid. Okay. Otherwise, in other characteristic feature, it resembles the viroid, HDV viroid. So how these viroids have originated? So many experts in this particular field, they believe that they considered these viroids are primitive viruses because they do not have any biosynthetic machinery. They are much primitive than viruses. Primitive when compared to viruses. So because of no biosynthetic machinery, no genes which code for protein or enzyme, so people believed that they are primitive viruses. But others believed that the present-day viroids are nothing but degenerative viruses. They developed from low molecular weight RNAs in the past and escaped from the cells, and they are still surviving on other host system. Okay, but some believed that they are abnormal RNAs. So nuclear RNAs might have escaped from the eukaryotic cells okay, and became pathogenic by mutation and causing diseases in plant. Somehow they are surviving. So these are the different, three different theories have been proposed. They are primitive viruses, they are degenerative viruses, they are abnormal RNAs. Okay, and Theo Daner, the man who discovered virions in 1971, in 1987, he made a statement. He hypothesized that virions may represent living relics. They are nothing but the remnants of the hypothetical RNA world. So before the evolution of DNA or protein, okay. So this is what Theo Daner, as in 1889, suggested, that they are the living relics of the RNA world. Okay, so we don't accept it that RNA world exists, because if you look at the central dogma, we believe that proteins led to the formation of the DNA. From DNA we have a RNA, from RNA to protein. So that's what we believe. But now recently the central dogma of theory has been modified. Now the RNA world does exist. There are different species of RNA molecules have been identified very recently. Lot of people research work has been done on RNA world, and epigenetic studies are being conducted in different parts, in plants and other human and animal system. The different types of RNA species have been isolated. Lot of work is going on in this RNA world. In that context, whatever Theo Daner stated, that there is a truth may be present, we have to investigate further on this. Whatever may be the our conclusions, virions are very curious, subviral, that means smaller than viruses, and they are known to cause serious diseases in plants, only in plants. They come under emerging plants. They are known to cause emerging diseases. So wild, where they are surviving, wild plants, whether they serve as a reservoirs of virions, we need to investigate wild plants also, whether any wild plants do harbor the presence of the virions. We yet to discover them. Very interesting thing is that minimal genetic information. They are so small RNAs, 250 to 450 base pairs, and made them very curious agents for study. How these small RNAs, they interfere with the host metabolism. Okay, host metabolism, hormonal imbalance, many because flower production, food production, it involves different types of hormone systems. So this hormone system gets affected. So how these small RNA interfere with the host metabolism, hormone production, all those things we need to study. Identification of disease is a challenging one. So powerful tools are now available, diagnosis and detection, and more studies are required to address issues related to mode of spread, agents responsible for the transmission of virions. Best way to manage viral disease by using phytosanitation and seed certification program is the only method. Diagnostic tools are available. Using diagnostic tool, we can certify that the seed material or plant material which we use for propagating purpose is free from viroid or not. That can be assured. So besides classical management strategies, biotechnological approaches, including developing transgenic plants against these viroids, gene silencing RNAi, gene silencing mechanism needs to be explored, and very interesting aspects are unfolding nowadays. So although the divide of mRNA viroid, they replicate autonomously and cause diseases in wide variety of plants. The lot of work has been done in the molecular biology of viroids in the last 25 years, but the precise nature of these molecular signals that are involved in metabolic disturbance, how they interfere, that needs to be understood. A series of questions first posed by Daner summarizes the many gaps in our current understanding of these smallest biological, unusual, molecular parasite. Lot of things need to unravel. So what molecular signals do viroids possess, or acellular RNAs possess? How do they interfere with the host metabolism, RNA polymerases, how mRNA synthesis gets affected? Okay, all these things we need to understand. What are the molecular mechanisms responsible for viride replication? Are these mechanisms operative in uninfected cells? If so, what are the functions? What determines viride host range? How they survive? Why other wild relatives, harbor this? Whether symptoms are also appear in wild relatives, we do not know. So all these things we need to understand, and most importantly, whether virides have other counterparts in human cells. Okay, whether animal virides are also present, that needs to be identified. So are virides restricted to only higher plant, or do they have counterparts in animal? So how did they originate? So that's also interesting aspect. Whatever we have only hypothetical and hypothesis, and that all these hypotheses we need to put together, and we need to understand many aspects of molecular biology, aspects of these, so that we can manage certain diseases. We can also use them biotechnologically for various applications like gene silencing and other mechanisms. So this is a small presentation, but very interesting aspects about virides. So end my presentation by just telling that this is what Louis Pasteur said, one of the great chemist and microbiologist of 18th century. What he has said, the importance of the infinitely small is infinitely large. It is very, very true now. Okay. So virides are very small, but their role is very bigger. Okay, small is okay, but they are playing a major important problems in agriculture and horticulture. Okay, so that is very interesting. So I have another few slides. Okay, so we can use total RNA extraction method. Lithium chloride method is the best method. To another one is CTAB method. The very recently trizol method is also available to isolate the RNA content from the infected plant. Okay, by using 2D page, we can separate the host RNA and also viride RNA. That is one method by which we can differentiate whether the host plant contain small RNAs or not. So 2D page will distinguish, okay, host RNA from the viride RNA. The other method by which you can, the same, once you have a RNA preparation, that can be used for reverse transcription polymerase chain reaction. By using that 1 microliter of the RNA, you can conduct a RT-PCR assay, convert the RNA, small RNA into cDNA, use that cDNA for further amplification and confirmation of associated virides, because powerful primers are available nowadays. So by using these primers, we can detect the associated viride by RT-PCR assay. Okay, so first PCR, and we have second PCR, two PCR, nested PCR, with both PCR and nested PCR. Very interesting thing that we can detect multiple virides by using different types of primers. From using the same RNA preparation, we can detect 3 or 4 different types of virides which are associated with a plant sample. Once you have detected, you can do bioassay by using the same RNA preparation. Okay, 1 microliter of the sample can be rubbed onto the bioassay indicator plant like cucumis tomato or cucumber or cucumis cucumber or tomato or Nicotiana benthamiana plant. And by radio, by doing that, we can double confirm the association of viride with any plant sample. Of course, now in addition to 2D page, we have hybridization techniques also available. So this is just to show that we have done some work related to potato spindle tuber viride in tomato. So this is a healthy plant. Okay, you can see how leaves gets curled here and distorted. Okay, they are all very well grown, but when it comes to flowering, forget the flowering, because the flowering will not take place. When there is no flowering, fruit setting will not be takes place. Though the plants are very lush green and grown very well, they are unable to produce the tomato fruits. So all these purpling, stunted growth, curling, stunted growth, purpling, epinasty, these are the important symptoms that we can detect. By visually we can, of course, these symptoms also overlaps with the virus diseases. So these are the important primers. So by using first PCR and second PCR, by using specific primers, we can amplify these viroids. And when you do that, you look at this. I told you the viroid size will be 250 to 450 base pair. Look at this. The size is only 360 base pairs in the first PCR. Okay. So you can detect the presence. When you use the specific viroid, you can detect the associated viroid. So you can elute this, sequence this, send it to sequencing. Sequencing will give you complete information about the complete viroid. So we can also conduct a bioassay. Even from bioassay plant, we can detect the associated viruses. You can see here, you can isolate the by lithium chloride method. You can detect the presence of viroid in all the inoculated plant by using the PCR and second PCR method. So this is how we can, comparison.
ಅವಳು ಸ್ವಲ್ಪ ಸಮಸ್ಯೆ
Siddesh
ಸರ್
sir,
Siddhesha sir, please mute your mic, sir. Siddhesha
Hello.
sir. Thank you, sir.
So look at this. This is one particular first paper, the occurrence of natural infection in tomato by potato spindle tuber viri in India. This is the first report we have reported very recently. Okay. A wonderful work on potato spindle tuber viri in tomato. Okay. We know that PSTVD associated with potato, but we have reported in tomato plant also. And molecular characterization. I told you it is a rod-shaped viroid. That particular structure we have also elucidated. Molecular detection and characterization of potato spindle tuber viroid in tomato. This is another paper. And as I mentioned, we did some work on grape. Bangalore region, they grow a lot of grapes for wine production. Okay. We have detected the yellow specks on the leaves. Look at this. Okay. So we have detected the association of GS grape, grape viroid. Okay. Grape vine yellow speckle viroid. Okay. Strain 2 in this Bangalore region. We have done the same. And look at this, the viroid that we have detected, it falls between 300 to 400 base pairs. We have completely sequenced it, and we have done phylogenetic studies, and we have yet to another interesting work that we have taken up very recently on molecular detection of abstent viroid. In the other part of the world, they have detected. We have for the first time proved that the abstent viroid is also present in mulberry. Look at top ones are healthy leaves, and the infected leaves, abstent viroid infected leaves will have the vein clearing, yellow speckling, leaf deformation symptom, and also mosaic symptom. These are the important symptoms, and we have detected the presence of 300 base pair length molecule, RNA molecule, and we have sequenced completely. Phylogenetic tree has been constructed. It was the viroid is 94% identity with viroid that has been detected in China and Japan. And this particular paper was also appeared as a first report of abstent viroid infecting mulberry in Karnataka. And these are some of the work that we have done. And this is my end of presentation. If you have any